Hello, listeners. Yamina here. Welcome back to the Dr. GPCR podcast. Today's episode is about a question that quietly shapes every GPCR program. Are your assays revealing mechanism or hiding it? My guest is Dr. Martin Marro, a cell pharmacologist whose career spans GPCR target discovery, signaling complexity, and translational decision-making across big pharma and biotech. When we recorded this conversation, Martin and I were actually working in the same building, a coincidence we discovered by pure chance. That's no longer the case, but the timing mattered, because this discussion feels like one of those rare, unscripted conversations where experience meets reflection. You'll hear how pathway selection changes interpretation, why some GPCR programs stall, not because the molecules are wrong, but because the questions are, and how bias, non-canonical signaling, and persistent signaling can reshape the fate of a program when the data don't line up the first time. Before we dive in, an important update for you. The Dr. GPCR University is coming back to the ecosystem. We're incredibly excited to unveil a new slate of courses launching this year, designed by scientists, for scientists, and grounded in real GPCR discovery challenges. We're also welcoming suggestions for course topics, instructors, and if you're teaching, building, or pushing the field forward, we do want to hear from you. All instructors and their immediate team receive a complimentary one-year premium membership with full access to every course, classified GPCR news, and more. And a quick thank you to our partners at Cell Research for enabling sharper GPCR discovery through fluorescent ligands and live cell assays. For more details and to meet the Cell team, links are in the show notes. And now, let's dive into the conversation.
Hello, everyone. This is Yamina from Dr. GPCR. And today I'm very excited to have with me Dr. Martin Marro. Martin, welcome to the podcast.
Thank you, Yamina. It's my pleasure for me to be here. Thank you for the invitation.
Thank you. This has been a long time coming. Lots of things have happened since my initial invitation. We even had the opportunity to meet in person, which I was very excited about.
Yep, that is correct.
So let's start at the beginning. Could you please introduce yourself? Tell us who you are and what do you do?
Yeah, so I'm the head of Cell Pharmacology and executive director at the Obesity Research Group at Eli Lilly and Company in Boston. So our lab was established last year. This is a young group of that, Eli Lilly has a large department working on diabetes, obesity, and complications, but most of the folks are in Indianapolis. So there's a new group that established in Boston a couple of years ago now, and my job is to establish a cell pharmacology lab. Start to recruit folks to join our lab, and that's what I've been doing. So our job is to develop in vitro pharmacology assays to perform both target discovery as well as drug discovery. So most of our assays are cell-based signaling assays, but sometimes we also run biochemical assays.
Oh, how cool. And actually, full disclosure, we work in the same building. It was completely by accident that we realized that we were working in the same building. That is fantastic. Yeah. Did you always know that you wanted to be a scientist?
I don't know, actually. I had some curiosity, when I was a child. I asked a lot of why questions, and my parents were physicians, and I had this, I was intrigued by the fact that a pill that would fit in the palm of your hand, could treat symptoms, and in some cases, cure diseases. So I think I had some hint of potential appetite for drug discovery, but I wasn't sure. I guess I was too young.
I love it. And when do GPCRs come into the picture?
Yeah, so when I was finishing my PhD, I was this is at the International Center for Genetic Engineering and Biotechnology in Trieste, Italy. So I was looking for doing a postdoc in all I knew is that I liked membrane proteins and cell signaling, but I wasn't quite fixated on GPCRs at the time. But it was very interesting. So I was at the library of this institute, and I was starting to apply for postdoc positions, and I knew that I wanted to continue my biomedical research, but in an applied setting. So I wanted to do drug discovery, and I was considering the possibility of doing a postdoc in industry. But I wasn't sure about that because I didn't know many people that had been through that path. So basically, I was at the library of the ICGB, and I bumped into this friend who worked at a different department, and he said, "Hey, I found this Marie Curie Industrial Fellowship to work at GSK on GPCRs," and that was the day before the deadline. So I applied for it, and I was lucky that I got it. So this is kind of the way I enter GPCRs. I was also applying for other postdocs in industry. I managed to get also an acceptance from Aventis Pharma in Frankfurt. That was, before they became Sanofi Aventis and then Sanofi. And I also applied for a few academic places in the US and the UK. So I was considering both industry and academia, but I was more interested in industry. And I think this was a fantastic opportunity to join a place like GSK in Stevenage in the UK to work in GPCRs.
Wonderful. I have two follow-up questions. Was it a long night to fill out the application since it was the day before the deadline?
It wasn't that long. I mean, it was long, but it wasn't that long. But
It was worth it.
It was worth it. Absolutely. Yeah. Every single line that I filled.
Wonderful. The other question I had for you, you mentioned in the beginning that you first got fascinated by signaling and membrane proteins. Do you remember, is there a specific time where you remember being exposed to membrane proteins and their signaling and saying, wow, I love this? Or was it just an accumulation of exposure?
Yeah, no, I think that happened during my PhD. So I was working on this cytoskeletal protein called adenosine, and we had knocked out the beta subunit of adenosine, and we found that those mice had a compensated anemia and also had hypertension, but we had no idea what the mechanism of action was. So we did a lot of nice studies in collaboration with Professor Screaming at UCLA, and we studied heart function, kidney function, and it was just hard to find the MOA. But one of our hypotheses at the time was that sodium potassium ATPase in kidney cells could be affecting sodium reabsorption and could explain, the mechanism of hypertension. So I was fascinated by studying, membrane proteins and sodium potassium ATPase, even if it's not a GPCR. That was kind of the first example that came into consideration, because I was trying to link this cytoskeletal protein cell signaling with that pump. So that was kind of I guess the conjunction between both adenosine and the pump that made me think that I think this is the way I want to go.
Wonderful. And so let's fast forward a little bit. You're accepted in this industry program, postdoc, which at the time, I think even now, a lot of academics don't know that that's an option, right? Because these programs are limited. I'm assuming they're also very selective. Yet, I'm curious to hear, how was that program in particular, and how different was it to do a postdoc in industry versus doing a PhD in academia?
Yeah, I think it was an excellent program. So at the time, Marie Curie fellowships had different flavors. This was an industrial fellowship. So the idea was that someone coming from academia would spend, would do the postdoc in industry, and the goal was to try and provide the knowledge of drug discovery to that candidate, but also allow her or him to conduct the research and being able to publish. And that's exactly what happened with me, and also with the idea of establishing collaborations as well with different academic institutions. So I was lucky to be the first. There were 5 fellowships for the same lab at GSK in Stevenage, and I was the first one to be accepted, and then others came. And the job was to do different projects, from target validation to expression and crystallography of GPCRs. Crystallography was very big at the time. It was way before cryo-EM, and some of my colleagues would try and express GPCRs and try to optimize expression of different GPCRs. I was fascinated about how much variation was in terms of the optimal expression time, or expression conditions between different GPCRs, even within class A GPCRs, right? Mm-hmm. And the idea was to for some of my colleagues to express and purify them to be able to crystallize them in a high throughput manner. My job was a bit different, so I was going to work on aptamers for GPCRs. So first of all, I think I was lucky to land in that place. The Steven site had a lot of GPCR talented folks. The biggest example that you and I know is Fiona Marshall. So she left that site the year before I joined as a postdoc. And, I worked with a number of colleagues that had been trained by Fiona or had worked with Fiona and had this fantastic academic profile, even within industry, right? So those guys, had an extensive publication list, including the cloning of the GABA B receptor, as you may know, the first demonstration of the GPCR heterodimerization, which was very novel at the time. They cloned the CGRP, the adrenomedulin receptors, they identified the RAMs, and they deorphanized at least 2 or 3 GPCRs. Long list of really, really good achievements. So I was very lucky to be surrounded by some of those folks. And my job was to basically work on aptamer discovery. I'm going to tell you in a second what an aptamer is, in case you don't remember or you don't know. And the idea was to basically identify these molecules for target validation purposes. So aptamers are small oligonucleotides, either DNA or RNA, that work like, they have antibody-like abilities, and they bind and block different targets, including proteins. So I was trained by Dr. Dion Daniels, who unfortunately passed away at the end of last year. Dion was a fantastic mentor and friend and taught me this SELECT technique for the identification of aptamers. So SELECT stands for systematic evolution of ligands by exponential enrichment. And it's a technique basically that allows you to identify them. So you have, first you start from a library of random oligos, really, really big library. And then you go through different rounds of binding, elution, and amplification until you find high affinity molecules. And then you characterize them, in cell-based assays, in terms of, cell signaling assays. So guess what? I ended up working with CXCR3, which is, I know, one of your favorite receptors. So my job was to try and find aptamers that would block the IP10 or CXCL10 binding to that GPCR. And I would also work on aptamers against CCL1 that would prevent binding to CCR8. So yeah, it was fascinating. We managed to get, really great molecules. We did a very elegant and comprehensive in vitro characterization, and which involved, multiple signaling pathways, including chemotaxis assays. And in the case of IP10, actually, we didn't, we actually went in vivo and showed activity as well in vivo. So, those aptamers had to be, of course, speculated to have, increase the half-life. And of course, they are engineered to, be resistant to nucleases. So anyway, beautiful work. I enjoyed that a lot. We managed to publish a couple of papers. And at the same time, that experience of 2 years allowed me to get to know what drug discovery was like. So it was really good.
Lovely, lovely. You mentioned chemotaxis, and I do remember how much I hated yet, and I learned to love how to set up chemotaxis assays.
Yep.
Initially, you had these glass chambers, and it was this complex thing. And I found this company that actually sold these 96 well plates, and there was a filter that you could apply. You had different well depths. I can't remember the name of the company, but it was fascinating because you could just, you could set it up. And I think I even have pictures of the droplet of cells that I would put at the top, incubate for 4 hours, and then collect the cells at the bottom, lyse them, and be able to quantify it. Just magnificent.
Yeah. That particular assay was run by a colleague of mine. So I would run all the other signaling assays, and he was an expert in chemotaxis. So David would run those. And it was fascinating. Yeah.
I think it's an art to be able to do that. It's absolutely different. And you mentioned also X-ray crystallography and the difficulties, even between Class A GPCRs, to find the right conditions. And I've told this multiple times during the podcast, and tell this to people: you have to get to know your receptor, and if your receptor lets you, shows you its needs, then you'll be able to make it speak to you or get a structure. But it's very difficult.
Yeah, I agree. And it's funny because things come and go in life, right? So now many people, including myself, are working with somamers, right? So somamers are actually modified, improved versions of aptamers, right? So these are slow off rate aptamers that are being used as reagents in this, these days for, they're basically designed to bind specific targets with high affinity and specificity, and you can use these reagents to as part of the SomaScan platform, that is a high throughput proteomic technology, right? So you can measure thousands of proteins simultaneously from a serum sample. So yeah, I'm still using them. So it's nice to know that Aptamers are still with me. Unfortunately, Aptamers didn't. In terms of therapeutics, there are only 2 FDA approved Aptamers. But we knew that was going to be very challenging. My job as a postdoc was to discover them for target validation tools at the time.
Oh, wonderful. So you spent 2 years at GSK as a postdoc. What happened next? Where did you go from there?
Yeah, by the way, I forgot to mention that also as a side project, I was exposed to different approaches to help my colleagues, my friends express challenging GPCRs, and we use different methods, including Backman, SFV, and others for structural applications. But anyway, so I had a successful and productive postdoc. I was very, very lucky and happy at the time. And my wife and I wanted to stay in the London area, and I started to look for jobs in industry that would meet my profile. And it was a hard time that year to find a job in industry. And I didn't want any job. I wanted to have a job that would allow me to grow in my career. And so that was around the time of the big exodus of big pharma companies starting from the UK, just leaving the UK one by one, which got exacerbated a bit later. But that year started, so there weren't many positions in pharma that would fit my profile. So I also reflected on the fact that my time in industry was going to come back at some point, and I had to be patient. So in the meantime, I joined a great lab. So I joined Hadrian Hobbs lab at the Wolfson Institute for Biomedical Research at University College London as a senior postdoc. And my goal was to expand my skills on receptor pharmacology. So Adrian had some collaborations with big pharma, which appealed to me as well, as I wanted to stay connected with the drug discovery world. So Adrian's lab was part of a bigger lab, which was led by Salvador Moncada. Salvador and his team were key in the discovery of nitric oxide as a signaling molecule. And that was kind of a pause from GPCRs for me, but I had a great time in that lab and I learned a lot. After 2.5 years, I had a chance to go back to industry by joining Novartis Institute for Biomedical Research in Horsham in the UK as a lab head in the gastrointestinal disease area.
Interesting. And when that opportunity came up to join Novartis as the lab head, was there a time where you were thinking, okay, so I spent time in industry, went back to academia, learned new skills? How did you at all think about, okay, after this position at Novartis as lab head, what else for you? Or did you ever think you'd be a professor at some point?
I don't think so. I fell in love with my job at Novartis, and I'm going to tell you a bit more about that in a second. I work with multiple GPCRs, and working with GPCRs in drug discovery is just fascinating, right? As you know, 1 out of 3 FDA-approved drugs target GPCR, and they're very popular and sometimes very challenging. So I didn't think that I was going to go back to academia and be a professor. I think that was kind of my starting point for my professional career. Even if 2 years before I had done a postdoc, this was my first, permanent job, and I loved it.
And so you joined Novartis as a lab head, fresh out of 2 postdocs, 1 academic, 1 industry. How did you go about, leading a team and building a team? Because that's a skill that, unfortunately, during graduate studies, they don't teach you that.
Yeah. So it was a gradual thing. So, during my postdoc, I had a chance to train a PhD student at GlaxoSmithKline for about 6 months, and also during my postdoc at Adrian Hobbs lab at UCL. I also, occasionally had to train, undergrad, but it was, temporary training. So this would become more permanent. So I was given a person, like weeks after I joined to be my first associate, and then a second person joined a few months later, and then other people joined my group in the following years. I had summer students, so yeah, and it was fascinating. I worked on 5 different GPCRs in 5 years. Of course, you run these projects in parallel in some cases. Some of them are well known and of public domain. So I work on 5-HT4. gradient receptor agonist, and others. And I learned a ton about receptor signaling, desensitization, de-orphanization, bias agonism, receptor kinetics, you name it. So I work on some characterization of tegasterol. Tegasterol is a 5-HT4 agonist for IBS. So I did some characterization, and that was one of my first projects. And then I also work on gradient receptors and others.
I love it. I love it. It's a whole different world, and I think from a, obviously you work at a big company, the direction and the targets, unless you're looking to validate a target, come from a higher strategic point of view. Yet you got to not only build the team, but also get to know those GPCRs and get a sense of what they were doing. That's fantastic.
I agree. And everyone is different.
Yes. Just as humans are different, GPCRs are also very different. And I think the tools also, the evolution of the tools plays also a critical role in better understanding GPCRs. And right now we're reliving a renaissance of GPCRs in drug discovery, which is, I think, a good time to be in the field.
Absolutely.
So after your time at Novartis as lab head, what was the next step in your career? Where did you feel like, okay, you needed to either learn more or take on more responsibility, or maybe, get more experience, either leading people or on a scientific level?
Yeah, so I spent 5 years at Novartis in Horsham. I'm happy to tell you a bit more about what I did, but so in those 5 years, I basically led to the discovery of several lead series for a challenging GPCR antagonist program. And then I work on receptor differentiation, leading to the identification of selective natural ligand that we were not able to publish at the time because there were more than one program focused on that receptor using different modalities. And I also did something that I'm very proud of, that I can cover this later. Was when you asked those questions about the aha moments that we had. But anyway, I was offered the possibility to move to Novartis in Cambridge, Massachusetts, to join the cardiovascular and metabolic disease area. This was after 5 years in Horsham, and I decided to go for it. I talked to my wife, and we loved the idea to come to Boston, and I joined the Cell Pharmacology group there as a lab head. And I was a slightly more senior lab head. I'd been promoted when I was in Horsham, so I joined that position. And then I was lucky that some of my former reports in Horsham came with me across the Atlantic, and came with me, and we continued working together. And then, my team started to grow over the years, and I became, I spent 10 years in that department. And at some point I was basically a senior group leader. and also in that department, I work with very exciting GPCRs, in this case for cardiovascular and metabolic diseases.
Hence the I think that's your niche at that point, where you got to this. And it's interesting because you mentioned doing your postdoc at GSK and Fiona Marshall having left GSK before you joined, and here at Novartis, I think you left and then Fiona joined the NIVR. So you've been, in a sense, missing each other on that front.
Yeah.
So we can jump maybe to the aha moments and then intertwine that with the next steps in your career. In the beginning, I'm really curious to hear a little bit more about those aha moments that shaped your career trajectory. One of them that stood out to me, maybe it's not part of those three, is the fact that you were first exposed to membrane proteins and their signaling, and being able to think about, okay, what is the MOA? Why do we see these effects, and how do you connect two proteins into a phenotype, basically?
Yep. So one of the aha moments was when I was in Horsham. So I was working on this GPCR, class A GPCR, were interested in agonist, and it was a GQ coupled receptor. So the team ran a high throughput screen, using calcium as a readout, and we had so many hits, and these were validated hits. They would, after the counter screen, a lot of chemical matter to play with. We ended up, improving, optimizing those hits with, the chemists actually ended up optimizing those hits. We had ended up with 4 lead series, and a lot of chemical masses to play with. And then we had, at some point, an ex vivo assay in the flow chart, and then we had, an in vivo mouse model. And, of course, in between the primary assay and the, ex vivo assay, we had a number of other, orthogonal assays, species selectivity or species cross-activity, receptor selectivity, etc. Typical assays that you would have in a flow chart or flow scheme for drug discovery. So what happened is that even if those molecules were from all 4 series were very, very potent in calcium, I'm talking about, sub-nanomolar, I should say, sub-nanomolar potency, in some cases between 0.1 and 1 nanomolar. In the calcium, they would just be, not very active in the ex vivo assay and completely dead in vivo.
Wow. And,
they had good PK properties, so we have no idea what happened. And we say, hey, this is a GQ couple receptor. We just cannot, connect, the in vivo data to in vitro. So, calcium being a calcium assay where it's very fast, we're reading at 3 minutes. Sometimes it's not the most predictive assay because it's far from an equilibrium situation, right? So at the time we're working with the IP1 assay, the HGRF format, and I say, hey, why don't we try and focus on IP1, right? It's more hemi equilibrium. You can actually play all the way to an hour incubation, even longer, and still be, in the linear parts of the standard curve and being able to trust your data. So that's what we did. And guess what? Same happened. So we just couldn't get, a good correlation with in vivo data. And so the chemists started to get a bit nervous, and the entire team was, we were scratching our heads because it was a high-profile program, and sense of urgency was a must-have. So the chemists proposed to do competition binding, going back to the old times where you would start, discovering drugs based on binding data. And we did a lot of competition binding. And again, it was just not predictive. And then I understood why that happened. But, my proposal as a junior lab head is to do 3 things. I wanted, first of all, to explore new pathways. So this receptor had a couple of publications that show that, that had shown cyclic AMP, potentially being linked to GS coupling, but it was very unexplored. I want to explore beta-arrestin. I want to explore bias agonism and also heterodimerization, and I'm going to tell you why. So, speaking of bias agonism, there were a lot of folks at the time at Novartis that were a bit reluctant to believe that all these examples in the literature about bias agonism were actually true bias agonism. Mm-hmm. So, as you may know, the concept started probably in the mid-90s, but took a while to come to terms and find real examples, right, within drugs, within drug discovery world. So, Novartis, imagine over 6,000 people working in research at Novartis at the time, a lot of people working on GPCR. So we decided to have a GPCR meeting, like a convention, right? Where we'll discuss a lot of things. And one of the topics was, let's redefine internally bias agonism. So the bar was very high. And, we would define bias agonism if you would have, let's say, compound 1 that was more potent or efficacious than compound 2 in assay A. Mm-hmm. And that scenario was totally reverted in assay B. Okay. Or if you have, that basically in the assay B, one of the molecules was death, right? Very, very extreme cases.
Yep.
And then heterodimization, the reason why I decided to study that, because it was reported for this receptor that it would heterodimerize. So the question came up, is it possible that we're looking at the wrong receptor and we screen against the wrong receptor? Maybe we need the elucidation, right? Yeah. So, did we look at the wrong pathway? Did we look at, agonists which are not biased? Did we look at the wrong receptor? Those are the questions that came on the table. And I wanted to explore, as an ambitious and young scientist, I want to explore them all. I had some pushback from management because I wanted to kind of pause the program. I wasn't leading the program, but I wanted to pause the program until we could figure out what was going on, right? But because it was such a high-profile project, and sense of urgency was a must-have, we couldn't do that. But in the background, I started to explore more the cyclic AMP pathway. We started to, we developed the HRF cyclic AMP assay. We started to test a lot of molecules in the background. And I persisted with my idea. And after a thorough analysis, we ended up showing, the data spoke for themselves. So I found that super agonism in the cyclic AMP assay would translate very well into in vivo efficacy, or would predict very well in vivo efficacy. And that was definitely an aha moment. So it was a long introduction, but it was important to see how my contributions managed to resurrect a project that was almost dead, and just trying to think outside the box and persist with my idea, despite having weaker potency. So the potency in cyclic AMP was rightward shifted compared to calcium, but this super agonism was kind of unique, right? So, and the reason why, the fact that the molecules were less potent than calcium is one of the reasons why I had pushback from management and say, hey, are you going to run a program on weaker potencies, right? But at the time we were trying to understand that actually potency was not as important as the Emax, right, as being super agonist. So anyway, the molecule reached the clinic, and again, is one of what I would consider one of my aha moments. The other one was when I was part of the team that managed to identify a natural ligand for this receptor I mentioned before. That was definitely a second aha moment.
Wonderful. Throughout the whole story you were describing for the first aha moment, I was trying to think, okay, which receptor would that be, and what kind of heterodimer? But I think it's such an interesting problem, right?
Yeah.
Because a program is designed, and people say, oh, you have to find GQ ligands. Then again, we don't know. And as Terry Kanakin would say, don't look for bias. Bias will find you.
Yep.
Because inherently nature is biased. And being able to drill down and figure out that it's cyclic AMP and the Emax, a super agonist, that gives you the phenotype or the therapeutic outcome is just fascinating. It's really awesome.
Yeah. And don't get me wrong, I benefited from fantastic discussions with other colleagues.
Of course.
In Horsham. So, we had in a different disease area, there was Stephen Charlton, Mark Dowling, David Sykes, and I would have, conversations with them, about pharmacology of the GPCR, but the cyclic AMP idea came from me, I have to say.
Oh, I love it. And I know David. David was also on the podcast.
Yes, I saw him. Yeah, I saw him.
A while back.
There were some very talented folks in Basel, people like Paul Soivan, who was the first one to report the pH sensing receptor, and there was a Nature paper in 2003. Fred Basilana, who de-organized a number of GPCRs, and then Roshi Buelal, who knew a lot about cell signaling. So I think being in Novartis, was for me also a huge honor to be surrounded by a lot of talented folks that, had a lot of experience with GPCRs.
Mm-hmm. Wonderful. So after your time at Novartis, I want to intertwine the inspiring young scientist part of the podcast with your career trajectory. So you were able to work on many more GPCRs at Novartis. The cyclic AMP idea. I mean, it's interesting because let me track back. I recorded a podcast episode with Dr. Michelle Hall. She's at Monash University, and she first got fascinated by the intricacies of non-canonical GPCR signaling, but her fascination started with cyclic AMP. Because you'd think, GS, cyclic AMP, and gone, but then there was a relationship between the GS and the GQ and the cyclic AMP and the calcium.
Yep.
And it's that balance that matters.
Absolutely. As you may know, calcium can come from multiple sources, including, type calcium channels. And so it's a kind of it's a less specific readout.
Yep.
So it's hard to tell whether that calcium we're reading was all coming from GQ as well.
Exactly. I was glad to hear that you mentioned the IP1 assay.
Yeah.
I feel like a lot of times the calcium assay is great because it's quick. You get a yes or no very quickly, but you absolutely need to follow up on that with an IP1 assay.
So I was telling before I moved to Novartis in Cambridge, Mass. I can tell you a bit more about what I did there if you want.
Please.
So I basically manage a group of scientists, MS or PhD level, a postdoc, and also summer students. We impacted multiple projects. as I said before, some of those folks have been working with me in Novartis Horsham before. And this is all public domain, there are patents out there, but I led the discovery of optimized GLP-1 receptor small molecule leads. These were agonists. I also was a key core team member on the discovery of a development candidate for another GPCR metabolism project. I'm a co-inventor of a patent where we discovered proprietary fatty acids conjugated to GLP-1. And basically, I studied the effect of receptor trafficking and of different agonists. So we basically extended the data that Stephen Bloom's lab at Imperial College in London had published on extending 4 mutants. This idea that if you have an agonist, a GLP-1 receptor agonist that preserves the stays at the plasma membrane for longer by being biased and having reduced beta resting, reduced internalization, and increased recycling, that would translate into potential better glucose lowering and also reduced nausea. So we extended that to our molecules, and so we use this DERET technology that is tear FRET assay. DERET stands for diffusion enhanced resonance energy transfer. We found that our proprietary fatty acid GLP-1 were less potent than semaglutide, which was the key comparator at the time in mouse and cynos AKMP assays. This is all in the patent. They were also weaker in beta arrestin, but they were more biased than semaglutide. And then not only that, but they also cause weaker receptor internalization and faster recycling than semaglutide. So the molecules were finding the profile we wanted, and then we found that they match semaglutide weight loss in DIO mice with less frequent dosing, and our front runner reduced body weight in a significant manner, being seno, and no emesis was observed. So that was really fascinating work that I managed to do. Again, extending what Stephen Bloom's had published with extending 4 mutants, we managed to extend that to more molecules and including ours. So it was very, very, very exciting. I also co-ran the global GPCR interest group at Novartis. As you know, many people working on GPCR. So I would co-run the global GPCR interest group. That was an initiative that would include, project updates and journal clubs, and I did that for 2 years. And I would also be an internal consultant on multiple GPCR projects.
Wonderful. And I think you mentioned having a convention across the company on GPCRs. And can you tell us a little bit more about how did that go and what were the take-home messages, if you can share from that meeting?
Yeah, so the idea was to discuss projects as well as new technologies and new trends, right? And bias agonism was one of them. That happened probably in 2007 to 2008. So as I said, there were a lot of people reluctant to believe that 100% of the bias agonist examples you would see in the literature were actually true biased. Could be system bias, could be a number of things, and most importantly, the implications of that, in physiology and disease. It was very hard. So that was definitely a point of discussion, and we decided to put a high bar in the redefinition, at least internal redefinition of bias agonism.
I think it's wonderful. All right.
And by the way, I managed to find some bias agonism in that project that I was referring to that transformed the one that with the cycling B pathway. Yeah, some of them were biased as well. Yeah.
I think it's wonderful. And as I was just mentioning, Dr. Kinakin always, whenever he teaches about bias, he says bias will do not seek bias will seek you, or something like that. And I think it's wonderful. It's just, I think in the beginning, we didn't have the right tools in order to assess the different signaling pathways, and also the mathematics to get rid of system bias and be independent of the cell line, independent of the experiment that you're looking at. And we definitely are there now. I forgot to ask you in the beginning, do you have a favorite GPCR?
So I don't have one favorite one. I like Class B GPCRs for obvious reasons. So they've been very hard to drug with small molecules, and it took decades for folks to tackle that issue. And there's still a lot of interest about them. And especially in the metabolism space. And I love orphans because, they're challenging. And, when you think that there are over 100 GPCRs which remain orphans, and some of them, have been attempted to be de-orphanized for decades. And it's just intriguing or fascinating at the same time that nobody was able to show what the natural ligand is. So, those remain some of my favorite ones. Yeah.
Wonderful. No, I agree. I think right now with the GPCR renaissance in drug discovery, the Class Bs are obviously at the top of that helm. So after your time at Novartis, what came next? What
I was
challenge
offered.
you wanted to tackle?
I was offered a job to lead the Cell Pharmacology lab at Tectonic Therapeutic, and Tectonic is a GPCR company co-founded by Tim Springer and Andrew Cruz of Harvard Medical School. So Tim Springer, he's a serial entrepreneur. He's founded many companies, Moderna, Seismic, Morphic. He's been very successful in doing so, and he's also an antibody expert. And Andrew Cruz is a GPCR expert. He did his PhD with Brian Kobilka, and the idea was to focus on GPCRs for biologics, especially on those difficult to drug GPCRs. So, I was very happy at Novartis after 15 years. So for me, it was hard to leave it, but I accepted that offer to join Tectonic. And, I was recruited as a senior director to lead the Cell Pharmacology lab. So we had to set up the lab and managed, and hard, great talent. And I was very lucky that I managed to get really, really talented people. I was also a member of the research leadership team in that company, and my lab contributed to the discovery of TX45. So we did all the cell-based characterization and signaling assays. So TX45 is a potential best-in-class Fc relaxin fusion protein. It's an RXFP1 agonist, heart failure with preserved ejection fraction. So looks like a great molecule. And in addition to supporting the development of TX45, I led the company's first GPCR antagonist antibody project from heat to a very advanced optimized lead. It is an anti-angiogenic, which is a potential first-in-class and also indication. And it was declared development candidate soon after my move to Eli Lilly. The molecule is called TX2100, and it's a BHH Fc fusion antagonist antibody that binds to an undisclosed GPCR target and that plays a key role in pathogenic angiogenesis. The focus is on an indication called HHT, which stands for hereditary hemorrhagic telangiectasia. It's basically the second most common genetic bleeding disorder. And the molecule is looking good. A surrogate molecule demonstrated efficacy in an animal model of HHT, and then a 4-week non-human primate dose range study with a functional equivalent precursor of this molecule. So no treatment-related toxicity in doses up to 100 mg per kg. I also try to, I love innovation, so I try to foster an innovative environment and creative environment. So I would lead the new target assessments and journal clubs, and also manage to, get multiple collaborations. So, and then a new chapter open to join Lilly, which is where I am right now, and I am enjoying it a lot. It is a good time to be in the obesity research at Lilly.
Absolutely. And while you were talking a little bit more about Tectonic, I think, I don't know if I mentioned this to you when we met in person, but I worked at the Institute for Protein Innovation, also co-founded by Andy Cruz and Tim Springer.
Interesting. Where's more work? I didn't know that.
Very, very small work.
I saw that.
I wanted to ask, going from small molecule to antibody discovery in the context of, running an in vitro molecular pharmacology lab and assays, were there any challenges? It's a new, therapeutic modality. And up until then, if I remember correctly, you're very much focused on small molecules and maybe peptides, but not necessarily antibodies. Were there any challenges?
Yes, absolutely. As you know, there are not many examples of antibodies against GPCRs, right? And it's always challenging. I mean, it only has a proprietary technology that looks very promising, but it can be challenging at times. And for me, I had to learn more about antibodies. So it's just a different modality, right? The way you define selectivity, the way you need to look at biophysical properties early on in your flow chart, the way you engineer them to improve, new concepts. Affinity maturation becomes the heat and lead optimization, right? But yeah, it was definitely a learning curve for me from that point of view, because I had done very little work with antibodies before.
Now you know. It's a whole different world.
Yeah.
Yet at the same time, whether it's small molecules, peptides, or antibodies, or all GPCR focused, for sure.
Yeah.
Amazing. Last question, which I think is the toughest question. What should we title this episode?
Yeah. So I didn't have a chance to tell you more about my history, but I wanted to call it From Green to Red, From Bread to Drugs via GPCRs. So I like it because it rhymes. But I didn't have a chance to tell you about how I started doing science. I actually started with transgenic wheat. So I moved from the green biology to the red biology. So yeah. So when I did my undergrad, I worked with transgenic plants. I was in Argentina, and we're trying to get transgenic plants that would increase the quality and protein content of bread. So these are proteins called glutenins that are expressed in the gluten. So we would, there was that dream that you would make, bread more nutritious and you would save the world, whatever, right? So, so yeah, I started working with transgenic plants, and that was nice for me because my grandfather was a baker. So I would make the connection that I would make better bread for bakers. But anyway, so that's why I call it From Green to Red. I moved from the green biology to the red biology, and from bread to drugs via GPCRs. So
I love it. I love it. I love it. You thought about it. I love it. Wonderful. I really love it. You mentioned your grandfather being a baker and also working on the, green biology. I worked for a summer at a bakery, and I find gluten fascinating. It is absolutely fascinating. I love baking. I try not to bake too much at home. When I do bake, I bake for the schools, the kids, and things like that. But I find it absolutely fascinating.
Yes,
that you can shape things, and you can make wonderful foods with gluten. I think it's just fascinating.
Yeah, and it's funny how these gluten proteins, the glutens and the gliadins, affect different properties of the bread. So that's really nice.
Yeah.
That was a learning point for me.
Yeah, absolutely. And how you can play around with different flours, different types.
Yes.
Spelt flour, for example, is one of my favorites, and it's more nutritious. It doesn't play around with your glucose index as much as regular flour. I think it's absolutely fascinating. If I had to pick another occupation, or maybe after a while, I might open a bakery. You'll be more than welcome to.
Please make sure you remember me. I'll be there for the inauguration day.
Absolutely.
I would like to give some advice to young scientists aspiring to contribute to the field.
Yes, please.
So, first of all, have fun. Find your passion. I think leave your comfort zone. I think it's very important that occasionally, once a week, once a month, once a year, you decide about the frequency that you leave your comfort zone. Yeah. There's this famous saying by Neil Walsh: "Life begins at the end of your comfort zone." And I think you can, slightly modify and say new ideas or discoveries may begin at the end of your comfort zone. So it's important you do that. Believe in yourself and don't give up in the face of adversity. Most experiments won't work. Most drugs won't make it. Occasionally, you may face setbacks as a professional or in your personal life. But the most important thing is that not what you face, but how you react to that, right? And if a door closes, a window or a bigger door will open. And then finally, build your network. So there's a nice book called Never Eat Alone by Keith Ferrazzi that I totally recommend it. It's important you build your network.
I love it. Actually, I've seen that book pop up. I have not read it, but I will definitely now look into it.
You have to. It's very good. It's going to change your life.
Wonderful. Wonderful. Thank you so much for the advice. I think it's important as well, as you mentioned. Most experiments won't work. And getting out of your comfort zone is essential for growth. You can't grow otherwise.
Yeah, I agree.
Thank you so much, Martin. Don't go anywhere.
Thank you for listening to the Dr. GPCR podcast. A huge thank you to Dr. Martin Marro for joining me. This conversation is a reminder that in GPCR discovery, progress often comes not from moving faster, but from asking better questions. His perspective on signaling, bias, and decision-making is one every GPCR scientist should hear. If this episode made you rethink assays, pathways, and how programs evolve, here's how you can help it reach more people like you. Share this episode with a colleague or a teammate, subscribe to the Dr. GPCR podcast so you never miss an episode, and leave us a review. It helps this show reach the scientists who need it the most. The Dr. GPCR University is coming back to the ecosystem. We can't wait to unveil the new courses launching this year. If you have any ideas for topics or instructors, we'd love to hear from you. All instructors and their immediate teams receive a complimentary 1-year premium membership with access to all courses, classified news, and all the premium perks. Thank you to Celtaris Research for supporting receptor science through innovative fluorescent ligands and live cell GPCR tools. For more details and to meet Celtaris Research, links are in the show notes. And thank you to the entire Dr. GPCR production team for making these conversations possible. Because receptors don't work alone, and neither should you. Until next time, stay safe.